Thanks, Manuel, for the introduction. Thank you to the organizer for the kind invitation. It's a real pleasure for me to be here today, and it's a challenge as well, because the talks have been so good so far. Quite impressive, actually, better than good. Several people presented the notion of computational modeling so far, and people mention as well as BML and the fact that we can encode those models. What I will present today is what we think is the next layer. In addition to present the structure of the model, we think now we need to put a bit more meaning in the structure of the models, and that's what I call the semantic layer. Most of you have seen such a diagram with the classical cycle experiment model, hypothesis experiment. And it's often represented that way. Douglas Kell already started to change a bit this representation, and I think the representation nowadays with the rise of systems biology is really the following, where the model is at the center of, it's not a cycle anymore, but the model is at the center of the scientific pathway, and you have your parameters, your experimental data. You design quantitative model. You then run simulation on various types of hardware, and then based on that you take decision, run other experiments, and you store the experimental data. That's functional genomics. What I would like to advocate is the fact that you need to store as well the models, because if those models have to be in the center of the scientific work, you need to exchange them, you need to record them for the future. So now we have a problem for quantitative modeling at the moment, and I think this problem is really the most important one we're facing, and the problem is the problem of the scale. And it's actually a multi-phase problem. We have a problem concerning the spatial scale. Most of people are developing model of signaling pathway here or gene transcription, but the notion of modeling in biology is much larger than that, and really it goes from the modeling of quantum event to the modeling of whole organism function or even population. So that's a large scale problem, but that's by far not the Okay, let's move on. That was funny. So back to the general introduction, so we have a spatial scale problem. We have also a time scale problem. I'm talking about the real time here, because molecular dynamics are at the level of the picosecond, and we go up to a life cycle of billions of seconds. But this is not the only time scale problem, right? We have also a modeling time scale problem, because here are different algorithms, and this is the real time simulated by 1 second of simulation time. And in molecular dynamics, you can simulate a picosecond in 1 second, while with continuous approaches and ODEs, you can simulate billions of years in 1 second if you wish. So this is a second time scale, right? So what can we do about that? Because obviously you won't be able to run everything in the same model, in the same simulation. So we can try to integrate models. So in this example, by some magic, we recognize that those two entities are the same, and we fuse the models. That's the first approach. We can encapsulate. So in the encapsulation, here we say this model actually can be summarized, abstracted into this entity. But all those are the same kind of model, like, for instance, kinetic model. What we can do as well is to communicate between various kind of data. So how can we do that? How can we communicate, integrate, and encapsulate? Well, what we have to do is build languages and methods to make things communicate. So we need standards of representation for all those, for models, for functional genomics data, for dynamic, new kinds of dynamic data set that are not generally recorded on a high throughput manner at the moment. We need to build interfaces, of course, and in particular, this is the work of ontologies, and that's a buzzword, but that's hugely important at the moment, not just in life sciences. And, well, we need to build even more data resources so we can store the things that are supposed to communicate. I will start with standards, and there is this famous saying that is attributed to almost everybody, which is the nice thing about standards is that there are so many to choose from. And people say that generally to boast about standard effort and basically sneer. But that's a very, very good thing about standards. A standard has to be perfectly fitted to the problem. It's not a problem to have a lot of different standards as far as they are clearly defined. And they do not, and they are not redundant. So we have a lot of standards of representation for quantitative models. The most known probably are CellML and SBML. I will talk a bit more about that later, but we have others in neurobiology. We have NeuroML, BrainML. You have languages like biopacks that are semantically rich but do not store quantitative information. You have graphical standards such as the nascent SBGN. So you have a lot of them, but by far the most successful has been SBML. So SBML is now kind of industry-grade standards adopted by a lot of software. At least all those software claim to be SBML compliant, and some are, in fact, pretty good at it. So it's been pretty successful. What is SBML for the one of you who don't know the beast? That's summarized here. The goal of SBML is to help people to disagree as precisely as possible. And if we achieve that, then we're very, very successful indeed. But more seriously, it's a way to encode quantitative model that describe compartments. Entities stored in those compartments with certain quantity, reactions between within compartments, between compartments. You can encode any kind of mathematical rules, and you can encode arbitrary discrete events that will affect the behavior of your model. So this is an SBML file, and it's obviously not important to see the detail, but across my talk I will use SBML quite a lot. So you define a compartment, species within this compartment, various quantitative parameters, and then, very importantly, you have mathematical equations here, and each symbol are taking the values of the entity they represent. And that SBML, obviously you will never see such a small SBML model, right? It's a fake model. But so far SBML has been described as to encode model of signaling pathways or metabolic network, but by far it's not restricted to that. SBML is meant to encode anything, any model, systems biology model, because system biology is scale-free. It's not just about molecules. And in particular, we have bits we call rich rule that can encode any rules, and we use it, for instance, to model neurobiological, electrophysiological behaviors. We have events, and what we call a species is not a molecule. It can be a molecule, it can be a cell, it can be an organ, it can be an organism. It can be anything for which you can give a quantity, amount or concentration. So the latest version of SBML is SBML Level 2 version 2 that's been released in September. And it's really, really a lot cleaner. It's more powerful. But unfortunately, it's very complex. And there is a message to take on. SBML cannot be written by hand anymore. It has to be written by a software. But it's very, very neat. And you have a nice specification. If you have some insomnia someday, you can. You need insomnia during 1 month to read it. But. The next level will be level 3. It's still a few years ahead, I think. But it will be modular, will allow to encode a lot more things like graph layout, generic reaction across multiple compartments. We will have mechanism to merge different models. describe arrays, describe the geometry of objects, their movements. So SBML is great, but is SBML enough? And if not, what's missing? Well, SBML is not sufficient to exchange model. It's a prerequisite, but it's not enough. Why? Because an SBML model lists the objects, but it doesn't tell us anything about them. It doesn't identify them in a way that is the same for me and you. And SBML model, an SBML model describes the mathematical expression that control the model, but it doesn't tell us what they are. It doesn't tell us this expression actually is meant to represent a Michaelis-Menten enzymatic reaction. You can parse the mathematical equation, but that will not tell you this is what was intended by the modeler. And of course, a SBML model is built for a certain simulation approach, and it's generally very hard to get from one simulation approach to the other just using SBML. So in summary, we cannot search easily SBML models. We cannot convert SBML model easily. And we cannot integrate them easily as well. We need something more. So the first step is to define a standard that tells you which information to add to your model to make it not only syntactically correct, but be more clear for the future user. And this is the purpose of Miriam, the minimum information requested in the annotation of biochemical models, but that's been forced onto me. I will say quantitative models. So here is the first table of flow. Your model has to correspond to the model described in the literature. So first, you need to encode it into a standard format, otherwise I cannot assess it represents correctly the model. It has to be related to one reference description that allow me to compare the results. The structure of the model must reflect the biological processes. This is very, very important. You can get exactly the same behavior with various different structure, as people who develop the oscillator know very well. And so you have various ways of describing a model that destroy completely the structure of the biological processes underneath, and this is bad. You should be able to run the model. If you click run and you don't get the right result, it's bad. It's not enough to have a model that apparently has a good structure. And because of that, you need to provide a lot of additional information that are generally not provided in the papers, such as initial conditions. So this is a small model. You have the biological structure of the model, the equations that describe the mathematical, the kinetic behavior of the model, but in addition, you have all kind of additional initial conditions. Second table of flow is the attribution annotation. You need to tell who designed the model and who is to praise or who is to be criticized for that. So you need to join the reference to the model, to the reference description. You need to add the name. You need to tell us if the model has been modified. And you need to tell people what to do with this model. Because a model is not a sequence. If you modify a model, it's not the same model anymore. It's not like a sequence. If you cut it in two, you have two sequences. If you pass them back together, it's the same sequence. So Miriam doesn't say all the models have to be publicly available. It just says that you have to precise what's the situation. And finally, you need to annotate those models in a way that anybody understands what are the components of the models and understand their biological significance. So if you have a model element and that represents a biological entity, you have to annotate it in a way that the relationship between the annotation and the model element is the same than between the biological entity represented by the annotation and the biological entity represented by the model element, right? And in order to do that, you have to use control qualifiers and control annotation. Obviously, the consequence is that the community has to agree upon a list of authoritative resources and qualifiers. So this is what we do at DBI. We have a database of data resources, databases or ontology, and So they are all stored, defined, and they have a standard unified resource identifier that allow to identify them. It's not a URL, right? ec-code.org doesn't exist. Just say this is the enzyme nomenclature. And for each entry, you have various information, an official name, synonyms, various form of the URI, definition, pattern to validate the identifiers, and various physical locations. Because one data type can be stored in various ways, in various physical locations around the world. And it's not up to us to decide which one is the right one. And obviously you have some documentation. The important bit is that we provide web services to access that. So if you go on the website, we have a web interface that is an example of the use of those web services, but we provide obviously the description of the web services, but also a library to implement an interface to Miriam. With those web services, you can generate correct annotation, and you can parse correct annotation and generate back the human readable stuff, or generate hyperlinks or whatever. So this is the same model than previously shown, but so this is the name of the model element, the URI that says this data is of type PubMed or this is a taxonomy ID, the identifier, and in addition, optionally, we put some qualifier that says that this model element is part of the biological process represented by the annotation. Okay, and this is the encoding in SBML. So this is a species called bound EGF receptor. Of course, I'm a human being knowing English language, so when I read bound EGF or EGF receptor, I can understand what the author meant by that. But this is by no means sufficient. So we have then the official annotation that says that this species is formed by two parts. It has two parts, and those parts are described by those URIs that point to a data type store in UniProt. This one is the epidermal growth factor receptor. This one, the epidermal growth factor. Now, is this sufficient? This is not. All that deals with annotation of the components, but it doesn't tell us anything about the math we use in the model. And here is a very practical example. You all know Michaelis-Menten, or so you think. Some people in the room knows a bit more and knows exactly what Michaelis-Menten is. And Michaelis-Menten is not what you are using all the time. What you are using is actually Briggs-Haldane. So this is the description of the math that everybody calls Michaelis-Menten. Actually, this mathematical expression has been proposed by three sets of authors, Michaelis-Menten, Briggs-Haldane, and von Slyke and Cullen. And in the three cases, the same mathematical expression represented different elementary steps, right? So one model says both steps are irreversible. Another model says you have a fast equilibrium between the enzyme and the substrate. This is Michaelis-Menten. And the third model says you have a quasi-steady state of the enzyme substrate complex. This is Briggs-Haldane, and this is what people use. Fine, we don't care as far as we use the so-called Michaelis-Menten. Now, let's imagine you want to merge two models, and in one situation, the enzyme concentration is fluctuating. Then you cannot use that anymore because you're not in steady state, right? Or this model has been generated for a continuous simulator, for simulation with ordinary differential equation. But I'm using stochastic simulation, and I want the elementary step. So I need to destroy this enzymatic reaction in two elementary chemical events. But how can I do that? Because the value of this parameter, K1, is different according to the formalism. So just giving the math is not enough. You have to give something more to precise the thought of the modeler. So one effort to add annotation is called the systems biology ontology. A quick word on ontology. So an ontology for today will be a set of elements of knowledge linked with sense-bearing relationship. there are hundreds of definitions of ontology, and I don't even want to enter the debate. The important point here is that each term of an ontology is associated with an identifier that is eternal. It will never disappear. It can become obsolete, but it doesn't disappear. This is very important, of course, because if you annotate a model, you don't want the annotation to become obsolete. And an ontology is an evolving structure, which is very, very good because we're in a domain that expands by the day. And of course, the ontology can be stored in machine-readable format because everything we do with ontology is done by computer. So we have 5 vocabularies: the type and role of the participant, like substrate, catalyst, but also physical things like macromolecule, channel, transporter, whatever. We have definitions of the parameters, Michaelis constant or forward unimolecular rate constant. There are specific parameters, each of them is defined. We have mathematical expressions like mass action kinetics or Henry-Michaelis-Menten equation, and those contain mathematical expression, and those mathematical expressions are annotated with the other vocabularies. So the parameters used in the mathematical expression have a precise definition. We store also the modeling framework to say if this model is supposed to be used in a continuous framework, in a discrete framework, in a spatial simulation. And finally, we have now a vocabulary of the event type, like catalysis, addition of a chemical group, control, whatever. This is the SBO browser. So here you have the example of the Briggs-Holden equation I mentioned before. This is a mathematical definition. It says that the semantics is linked to this modeling framework. Here, this is continuous framework. And this K-cat is actually the catalytic constant defined here. This substance S is the substrate that should be defined here. There was a change of resolution. We distribute the system biology ontology in various formats. OBO, the Open Biomedical Ontology format, OWL, the Web Ontology language. And as previously, we distribute, we have web services that allows you to access SBO, retrieve a term, retrieve a definition, retrieve an ID, do something with that, with the library provided as well. So you're welcome to contribute methods if you want us to add methods in those web services. Oh, good. So now we have our model of Michaelis-Menten, and we can annotate all those in SBML, specifying, putting some meaning on those species. So A will be a substrate, B a product, C a catalyst. This kinetic law is a Briggs-Holden equation, and those two parameters U and V are Km and Kcat. And that emphasizes what I said before. Yeah, if I say Kcat, you know what I mean, but if I say V, who can guess I'm talking about Kcat, right? But the computer will call it V, or even worse, VXY0001. So now I have this, and if I have a continuous simulator, I can generate this rate law. If I have a discrete simulator, I can generate those 3 different rate laws. Of course, I have an unknown variable here. I will have to do some parameter scan, but at least I can do that now. And it's even more. I can have the complete model. And here I annotated the species definition as well. And I know that those two are simple chemicals. This one is a macromolecule. And this reaction is a catalysis. So now I will be able to link that to a graphical standard, for instance, to represent the various bits and pieces with specific glyphs that are standardized as well. Okay, so we can link SBML to a database of biological information using Miriam, and we can link the various standards with SBO. Now we're ready to exchange, but in order to exchange, we need to have things to exchange. So this is the idea of the unified model resource and biomodels database. So a unified model resource has to be atopical. It should not be tied to a specific biological substrate or a modeling approach. It should be a real searchable database, not just a web page with link to models. And of course, we should verify the structure and the meaning of and the results of the simulation instantiated from the models. It has to be obviously developed as an international collaboration. It needs to be freely available and reusable. That's not Miriam, right? That's biomodels database. And of course, it has to be a long-term effort. So this is biomodels database. As I said, we store and serve quantitative models only of biomedical interest. Often people ask us to store things that have no relationship. We store only the models described in peer review literature because we don't want to mess up with the validity of the model. If a peer review accepted the model, that's good enough for us. And all the models are curated. They are simulated, so we know they're working. The components are annotated, and we distribute them, of course. So this is the structure of biomodels database, but I will go a bit faster here. You can search the database. So here, for instance, I want to search the database using gene ontology term, and I put mapk. So mapk is just an acronym. Some of you knows that it's map kinase. A smaller subset knows it's mitogen activated protein kinase. But of course, you want to search using those three terms and get the same result. So we search using the gene ontology annotation of the models, and we retrieve the models here. So all those models deals with map kinase cascade in some sense. Some are obvious because it's in their name. Some are less obvious, like a model of yeast pheromone. So once you selected your model, you can access a small web interface that tells you information about this model, allows you to link to other databases, and you can access graphical view of the model, and you can check that we actually did our work and simulated the model and check that it worked. Finally, you can download the model for your own consumption in various formats, and in particular in SBML, and then you can run it. So that's very, very, very important. That was not possible a few years ago, and that's one of the three Popper's criteria for scientific activity, right? Reproducibility. If you cannot reproduce something, it's not science. Well, I tell you, until 2000, you had no modeling that was science. So here, I have the same model that I uploaded in Copazi and Cell Designer, and directly run them. I uploaded the SBML, and here I used export from SBML converted to XPP and CLab, and I get the same behavior, which is good. So where are the models coming from? They're coming from existing model repository. We swallowed the whole SBML repository. We swallowed the part of GWS Online in SBML, and now we have an agreement with those databases, and we are progressively acquiring all their models. But now, most of the models are actually coming from you, from authors submitting papers or asking asking grants and needing accession number for the grant. But they are also forwarded from journals, molecular system biology, plus computational biology, and now BMC systems biology as well, I think. And of course, we encode ourselves new models because to curate model is very, very tedious sometimes. So to have a bit of pleasure, we encode new models. And this is the increase. Well, classic, like every database in life science, we bumped into the exponential growth now, which poses significant problems, but this is really, we're happy about that at the same time, because it's more work, but it shows that the database took off. And it also shows that people are generating more models that can be encoded in SBML. Another thing you can get from this graph is that the models are bigger and bigger, because here you have the number of reactions, so we are about a bit more than 6,000 reactions now, and you see that the ratio of reaction over models is increasing. You have more and more reactions per model. Lambda phage is finished now. We are modeling bigger things. As I said, we are pretty atopical. And this is a complete mosaic. So biomodels, the various standards, SBML, BioPax, SBGN, and then we can link everything using the annotation, the ontologies, and hopefully we can link the experimentalists to the modelers in the case they are not the same person. This is the biomodels.net team. In particular, Camille developed the Miriam resource, Melanie Courteau developed the SBO support, Barco Donizelli and Chen Li are developing the core of biomodels database, and those people are annotators. But the biomodels.net effort is something much larger, and in particular, it's developed in collaboration between DBI and the SBML team in Caltech, but we also collaborate with many other teams, like the team of Herberto, the team of Upindorbala, and the team of Kitano, JWS Online here in Manchester, or partially here, and the community. So before, like 4 or 5 months ago, I had a big list of people here, but now the list grew out of the slide, so I'm sorry I can't acknowledge everybody, but of course it wouldn't be possible without all the people contributing to SBML, contributing models, criticizing the annotation and things like that. And I'm done.
So that was great. I'm sure that you'll have questions, especially for those who were doing modeling before 2000. And
I would.
And please, if you have some questions. I have one. What is the difference between the first version of SBML and the version number 2? What have been the improvements? And what have been the lessons learned, the errors, and the sort of ideas?
The big difference between SBML level 1 and level 2 is that SBML level 1 was developed to cope with existing simulators. The purpose was to have something working really quickly that could be adopted really quickly. So we were not really picky about the structure, and we didn't know about it. I mean, we were new to XML. Well, XML was new. And what level 2 is completely different. Level 2 is much more structured, much more agnostic in terms of software, and it's much more robust. Another thing we learned developing the various versions, not the level, is the backward compatibility is absolutely crucial. So now every change of SBML is really has tremendous effect on the community because so many people support it. So we're very, very cautious about adding or any feature, or even worse, changing the meaning of things, which explains the delay between versions that is progressively increasing.
More questions?
Okay. When do we pass modeling some diseases?
There are models of diseases in biomodels database.
Really?
There are, in particular, there is a model of cancer of detoxification of anti-tumoral agents. It's up to the submitters, really. So I hope we will have more in the future.
So my question is, why are there multiple versions of the same model for the same paper actually?
Yes, that's our definition of a model at the moment is driven by SBML. So this is not the generic definition of a model most people have. As soon as the structure of the network is different, we consider the models are different. And as soon as the qualitative behavior of the models are different, we consider the models are different. Meaning, if a model with a certain value of a parameter oscillate and with another value doesn't oscillate or has a chaotic behavior, we will store 3 versions. If this is an amplitude of oscillation that changes, we will put a note saying if you change the parameter, you will get. If there is one extra reaction, we store 2 models. we hope that with SBML level 3 we will be able to store ranges of parameters, and then we will fuse back all those models together. It's, yeah.
More questions?
Yeah, just one question. You've stressed the importance of reproducibility of the models. Is it more than a snapshot that you're providing? Are you giving numbers so that people can really validate what the simulator is doing?
Currently, it's snapshots. We are, as you maybe know, the SBML team is developing something called the test suite that will test, allow to test simulators. Once we have that, we can use the mechanism used in the test suite to validate any models. But what we will do in the next version of biomodels, I mean the next, not release, but version of the software in 1 year, is to provide a very simple online simulation system. Nothing fancy like JWS Online, but where you will click and with SBML OD solver have the curve and the numerical output, like you have on the online version of SBML OD solver at the moment. So you won't be able to change parameters or things like that, but you will be able to get the values. Yeah.
One more question.
I was wondering, with multimeric enzymes, where you often have a lot more details to do with how many enzymes there are, and you get the cooperativity, would the ontology include terms for that, and to provide all the details as to, because they're a lot more complex and diverse, the actual equation forms for multimeric enzymes. So would that actually be possible as well?
What we started to do was to store raw rate equations, and we bumped into a similar problem immediately. You have dozens of forms for the same equation. So now we have mathematical expression in the parameters as well. So then we will not enumerate all the form of Michaelis-Menten equation, for instance. We'll have a generic form and then different form of parameters, and it will be up to the user. For allostery, we intend to enter all the possible different rate laws, but this is an ontology. This will be driven by the users. If nobody asks us to encode this particular funny rate law, we will not. We have better things to do. But if somebody asks us to enter this law, we will add it. Yeah. So I guess biomodels database will be the main driving force once we start annotating SBML with SBO terms.
Right, thank you.